to take a quick look at how T cells develop into the helper T cells and the cytotoxic T cells. In other words, we want to talk about the development of T cells. The progenitor cells that actually form T cells are from the bone marrow and migrates from the bone marrow to the thymus, and that's why they are called the thymocytes. And it is these thymocytes that develop into mature T cells. And these progenitor cells, called thymocytes in the thymus, are called double negative thymocytes because they lack the common CD4 and CD8 markers. So they lack those markers, so they are called double negative thymocytes. But then there are two different types: those who have CD25 and those who have CD45. But generally they all have CD3. So we have those who have CD3 and CD44, and those who have CD3 and CD25. Now, the double negative thymocytes with CD25 undergoes beta selection, while those who have CD44 die by apoptosis. And what is beta selection? This involves the pairing of the beta chain with alpha chain to form the T cell receptor. And then these T cell receptor form a complex with CD3, and then you have a differentiation that develops into the appearance of CD4 and CD8, which act as T cell co-receptors. So in beta selection, beta chain pairs with alpha chain to form TCR. And this TCR, which is the T cell receptor, form a complex with CD3, which makes the cell to differentiate. And what you have next is the appearance of two major markers, CD4 and CD8. And these are what act as co-receptors. Mind you, these double positive cells are non-specific because they contain both the CD4 and the CD8. At that point, they have to interact with the antigens. And so, when they interact with the antigens, and then you have another selection, antigen affinity selection. Now, those who interact well with the antigens are spared, while those who don't interact well with the antigens, they also die off. So they bind to the antigen via the MHC1 or the MHC2. So those that are spared are those who bind well with strong affinity. After that, you have the downregulation of either of the co-receptor, either downregulation of CD4 or downregulation of CD8. Now, those with downregulation of CD4, you have CD8 single positive cells, and those with downregulation of CD8, you have CD4 single positive cells. So that is how you have a single positive cells. The CD4 cells are known as the helper T cells, while the CD8 are known as the cytotoxic T cells. And the CD4 is a T cell receptor co-receptor, which help TCR or the T cell receptor in communicating with the major histocompatibility complex or the MHC2. I mean the molecule of antigen presenting cells. Before this interaction, the T cells are naive, but after the interaction of the T cells with the MHC molecules, then the TCR undergoes a configurational change at the variable regions, and then the T cell is no longer naive, it is now specific for that antigen. And this is what we mean by T cell activation. And once T cell is activated, it produces cytokines. And these cytokines interact with cytotoxic T cells, interact with B cells, interact with phagocytes. In other words, they are activated by the antigen presenting cells, and then they in turn activate effector cells, and the effector cells are those listed: cytotoxic T cells, B cells, and phagocytes. Thank you.